Welcome back, everybody. My name is Stephen Smith. I'm one of the two Stephen Smiths in the room at the moment, I believe. And it is my great pleasure to introduce Dr. Michael Yartsev, a new professor at the University of California, Berkeley. We're heading into the stretch. This is the last of the non-lightning talks. And I would like to say we saved the best for last. But of course, that would be completely silly, as you all know. Forget about all of them were above average. All of the talks have been the best so far, don't you think? Yeah. But I just spent a few minutes talking with Michael, and I know for a fact that you are in for a real treat. We did save something completely different for last. I think that is safe to say. So Michael has already had a very distinguished career, judging by an extraordinary number of awards, which I'm not going to attempt. I tried to memorize them this morning, but I gave up. But a couple that stuck in my mind were a Kanishi Neuroethology Award. I'm sure that meant a lot to Michael. And an Eppendorf Grand Prize, and that probably meant a lot to Michael's bank account. So I would like to go on, but I don't want to deprive you of an exciting minute of the completely different thing that Michael is going to tell you about. So I think I will just sit down and get out of the way and welcome Michael up to the podium.
Okay, thank you very much. It's good? Yep. Of course, I want to start off by thanking the invitation to come here and the option of being part of this amazing organization. This conference has been very interesting so far. Interacting with all of you was really exciting, and I look forward to interacting more later today and tomorrow. So indeed, as you mentioned, thank you very much for the introduction. Can I actually ask to turn those lights off? So it's going to be just a small movie. Is that possible or not? It's not a big deal.
There's a camera on you. We need to see you.
Okay, fine. No problem.
Light pollution comes from the windows up there. So.
No, that's fine. No problem. So yeah, indeed, I work on slightly different things and animal models, these guys. And I don't work on vision, I work on mice, but I do use bats to address questions which are core to all mammals on the planet, and specifically questions which are very difficult or potentially even impossible to address in other mammalian model systems, which I think bats are such an exciting model system. And specifically, my lab works on two main lines of research. The first one is the neural mechanisms of spatial behavior, which I'm going to talk a lot about today. And then we go on a completely new and unexplored line of research, new line of research, the neural mechanisms, social communication, and vocal learning in bats. And I'm going to talk towards the end why I think that's so exciting. So today I'm going to tell you mostly two stories on the work we use the bats to study spatial representation in the mammalian brain. First of all, in crawling bats on the entorhinal cortex grid cells, and then in freely flying bats, talking about 3D spatial representation. But before that, and specifically in the context of spatial representation and spatial behavior, I'm sure many of you are sitting here in this room and you have one major question in mind, and that is probably the question, why bats, or perhaps even why the hell bats? And that's, a very valid question, right? As we saw, we have the mouse model system, so it's beautiful behavior, genetics. Why the hell would you want to work on this unusual mammalian model system? And so to begin addressing this question, instead of just explaining that, I'm going to invite you to take a little flight with me and with the bats. So this is a work done not by me, by Asaf Tzohar in the laboratory of Nachum Lanovsky. And so there's no neural recordings just yet. But the remarkable thing Asaf did is that he put tiny GPSs on the backs of our bats, Egyptian fruit bats in Israel. And this allowed them to track the natural navigation of those animals in their natural environments over hundreds of kilometers or tens of kilometers. So what you're going to do now is take a little flight with the bat. So this is a particular bat cave in Israel. The red trajectory is going to be the flight of the bat. This is, of course, done during the night, just overlaid on a Google image during the day. And so here you can see the bat taking off, and it starts navigating towards his most favorite fruit tree, which is located over all the way at the end over there. And one of the first things you can notice about this behavior is just how straight the flight trajectory of these bats is. And so they've shown very nicely in this study the bats actually use local cues to navigate in the environment. This is the main highway in Israel, Route 6. And they follow these routes again and again, night after night, going back and forth very reliably. They use local cues. So here the bat is going to fly along the backyards of this little kibbutz. He's going to make a little stop at some point, and then he's going to come to the Air Force Base, take a right turn at the Air Force Base, and then it's pretty much smooth sailing to his most favorite fruit tree, where he's actually foraging for the rest of the night. So one of the simple motivations, why would you want to use a bat to study spatial navigation? Well, that is because these animals are incredible spatial navigators with something which we cannot say for mice and rats, and it's potentially a good idea why you want to study them. I'm going to give more motivation as we move further on through the talk, why we use a bat for the specific questions I addressed. But before I go to the bat, I want to come back for a second to the rat, because the entire work in the field on place cells, grid cells, and the spatial navigation system was, of course, which was awarded the Nobel Prize this year for in physiology and medicine, was done exclusively in rodents. So I'm just going to describe the main components or the main characters of today's story. So the first one is the place cell discovered by John O'Keefe in the hippocampus. And you can see the activity of one such place cell over here. In black is the running trajectory of a rat as it moves around in a square box. The red dots are action potentials emitted by a single hippocampal neuron. You can see most of the action happens in this restricted region of the environment. You can also see it here below in this color-coded firing rate map. So the blue means zero firing rate, and the red means maximal firing rate. You can see this area is where most of the activity occurs. It's called the neurons place field. And then in 2005, the Moser group, Edward and May Breed, went one synapse back into the main input structure into the hippocampus, where they discovered this beautiful grid cell, which again fire in a very tight relation to the animal's position in the environment. But as you can see, there are many of those activity fields, and they're called grid cells because all of those activity fields occur on the vertices of an hexagonal grid, which then continues and spans the available environment in the room. In the medial entorhinal cortex, they also describe head direction cells, kind of the compass of the map. For example, this is one neuron firing whenever the animal's head is pointing towards the east, and the border cells which fire when the animal is near the borders of its particular environment. So I'm going to talk about all of these spatial cell types today, but the main kind of characters will be the place cell and the grid cell. And so the first project that we have done, where we thought we might be able to use the bats to do something interesting, was actually trying to address a question that was defined by many as the most basic and fundamental question of all about this grid formation, and that is, what are the neural mechanisms that give rise to this beautiful firing pattern? And the nice thing is, since the discovery of grid cells in 2005, there has literally been just a flood of computational models coming out trying to account for this grid formation. And all of these can be classified as belonging to one of two major classes of models. So the first one are network-based models, which, as the name implies, rely on the network of the entorhinal cortex area, and possibly even outside of the entorhinal cortex and the inputs into the entorhinal cortex. And I'm not going to be talking about those models today. I'm only bringing them up because these are the complete contrast of the second class of models, which at the time were the most prominent class of models in the field. And these are called the oscillatory interference models, which are just a single neuron model. And basically what those models say is that this firing pattern is generated by an interference between brain oscillations, in kind of proposing a transfer from oscillations in time into oscillations in space. And the idea behind them is very simple. If you have the membrane potential of the neuron oscillating at a particular frequency, as you will see in a second, it's going to be the theta frequency, 4 to 11 hertz, which is very prominent in rats. And you also have a dendritic input arriving into the neuron at a slightly shifted frequency, then those two oscillators, or more oscillators for the two-dimensional space, are going to basically work together. And when they're out of phase, the neuron is going to be inhibited. It cannot spike. When they're going to be in phase, the neuron can spike. And if you then add multiple of those oscillators, you get the grid formation. And I'm going to lay out the prerequisites of those models in the very next slide. But first of all, I just want to explain to you again the rationale of why the hell would you want to use bats to address this mechanism, right? And so the logic was very simple. So in rats, which is the only model system at the time where those grid cells were reported, you always find simultaneously the existence of the grid cells and this continuous form of theta oscillation. So you can only look for correlation between the two, but causality was essentially impossible to examine. However, in the bat, and this was done by my former mentor, Nachum Lanovsky, and then I also found this in our bat species in Israel, you can find perfectly beautiful place cells or place fields to exist in the CA1 hippocampal region of bats in the absence of these continuous theta oscillations. And specifically, we found that theta oscillations appear in these very short and intermediate theta bouts, very similar to the way that these oscillations exist in primates and in humans. So basically, we thought, how about if we now find, but there was no like a clear hypothesis about the place, but we thought, how about if we now find these grid cells to exist in the bat, but in the complete absence of the main prerequisites of those models? And this was precisely what we set out to do. So it's kind of an unusual logic, right? But it says perhaps studies in bats could allow causal examination of the most major class of models, which is based solely on data from rats. And that's what we set out to do. So just to lay out the main prerequisites of those models. So the first one is that you should have this continuous form of theta oscillation in the entorhinal cortex. And the continuous notion is critical because according to those models, if the oscillations are not continuous, the grid formation should evaporate. And of course, this is motivated by data that was known before in rats. And you find that if you go into the entorhinal cortex of the rat, the entire power frequency display is just dominated by this massive peak in the theta frequency band. So that's prerequisite number one. And the second one is that if the firing pattern of the grid cells is generated by an interference by oscillations in the theta frequency band, then by definition, the firing of those neurons should also be modulated in that frequency. And this is exactly what you find, of course. This is an autocorrelogram of a single entorhinal cortex grid cell. You can see this very beautiful modulation every roughly 120 milliseconds, which is 8 hertz, and that is exactly theta oscillations. So those models were driven by this data, and of course, henceforth supported by it. So we set out to examine those models in the bats as well. So we collaborated with Menno Witter, who delineated the entorhinal cortex of the bat for us. I trained bats very painfully for many months to crawl, and that actually taught me a very good lesson not to go against the natural behavior of the animal, but we had to do it because we wanted to do the experiment in exactly the same way as it was done in rats. So bats are crawling in these large boxes. And sure enough, we found that many of the cells were bona fide grid cells according to every possible measure we could examine that was, of course, derived from analysis in rats. We found many, many such grid cells, either with head directional tuning, known as conjunctive grid cells, or without head directional tuning, called pure grid cells. We found also the same head direction cells. We found the same border cells. And these were distributed throughout the different layers of the entorhinal cortex. I'm not describing it here in the interest of time, but we carefully characterized all the properties, the known properties of those grid cells, and we found them to be absolutely identical to all of those that were reported before in rats as well. We looked at brain oscillations, and we found exactly the same brain oscillations you find in rats, in primates, and in humans, also to exist in our bats. So one of them is high-frequency repole oscillation, occurring at about 120 hertz. And we also found gamma oscillations, so low gamma and high gamma oscillation. But there was just one single difference that we could find between bats and rats, and that was exactly our prediction. So we found that in the bat, theta oscillations were by no means continuous. They appeared as we expected in the form that's very short and intermittent Theta bouts usually occur in under 1 second and often just minutes and minutes apart. If you look at the power frequency display during behavior, low velocity, high velocity, sleep, doesn't matter what reference you basically use, you will never found this peak in the theta frequency band. And if you basically look at the autocorrelogram of the bad grid cells, you see there's no periodicity. If you don't believe me, this is the FFT of the autocorrelogram. You can see there's absolutely no power within the theta frequency band. So we basically found perfect grid cells with exactly the same properties as you find in rats to exist in the complete absence of all the prerequisites of those models, and that allows us to causally argue against the link between those oscillations and the grid formation, or at least against the generality of the rodent finding to other mammals as well. Now, this was great. But then we realized we were basically doing things which were very similar to what you do in rats. And the way that we were looking at the grid formation is kind of like the way that everybody else were looking and how you would look at the box of oranges if you walk around in the supermarket, which is basically a two-dimensional view from the top. But all of us live in a three-dimensional world, right? And looking at this kind of an image doesn't tell us how those oranges are actually arranged in 3D space. And exactly the same question can be applied to place cells. So when I recorded this one single hippocampal place cell in the bat, in the crawling bat, this tells me very little about how those firing fields would exist if they were to be examined in the true 3D space, because really all I'm doing is taking a single cut through the three-dimensional plane. So that basically led to the second project, subsequent project, where we had to develop the technique to record wirelessly from 3D flying animals, something that was never done before in neuroscience. And people, I'm not going here into the details of it, but before people try to use rats to study 3D space, and I think that's an example of why you should not use one model system to address all questions in neuroscience, but try to choose the appropriate model system. And they made really sincere and difficult attempts, like having rats move on these Guggenheim-like mazes, or perhaps more extreme measures, like sending rats to outer space. But and that I can discuss the details of this if you want. But of course, our approach was saying, let's use the only flying mammal on this planet to ask how is 3D space being encoded in the hippocampus, and that is, of course, the bat. So at this point, I already learned my lesson not to go against the natural ethology of the animal. So we basically built this artificial fruit tree. What I didn't show you, when the bats reach the fruit tree at the very end of this movie, they just go forge like crazy around that fruit tree, for a while, cover the entire 3D volumetric space around it, which is precisely what we wanted to study 3D encoding. So we just built the artificial tree, and as you're going to see in a second, you get beautiful 3D behavior from the bats. We track at the time their position using two video cameras. We had a telemetry system. We have the antennas on the side of the room and microphones to monitor their echolocation. This is about a 6 by 5 by 3 meter flight room, similar to the one we're building now at Berkeley. And this is an example of a neural trace recorded from one single tetrode implanted into the bat hippocampus. This is during a particular flight trajectory of the bat. The pink portion shows the time during which we recorded this activity. So you can see you get very nice SNR, very large spikes. I wanted to emphasize that it's not all roses, because sometimes you do get these wing beat artifacts on the first days of recording as the bats are hitting the telemetry system. But then within just a couple of days, they learn it's not very pleasant for me to be hitting the telemetry system with my wings. So they stop doing this, and you basically get a perfect clean signal like this with spikes, and you can isolate the individual neurons using just standard methods of spike sorting. So now that we have those two things together, a true three-dimensional volumetric behavior by a mammal and the ability to record neural activity during this behavior, we could address for the first time how do hippocampal place cells encode 3D space. And here is the answer. So you can see here again in gray the flight trajectory of the bats. This is one single bat. This is the red dots at the action potential of a single hippocampal neurons, the waveforms of which you see over here. You can see most of the activity is localized to a particular region of the environment. Here on the right is a color-coded firing rate map of this particular neuron. Again, from blue to red, you can see again most of the activity is here. And you might notice that those firing fields look suspiciously like balls, right? Effectively, balls hang in mid-air in 3D space, and that was exactly the first finding. So those firing fields were essentially perfect 3D isotropic balls, meaning those firing fields were equally sensitive to all the different dimensions and axes of the environment because they represented all of them with a similar resolution. When we looked at the collective activity of multiple of those fields in different bats, so each of those convex holes represent individual place fields. This is bat 1, bat 2, bat 3, bat 4. We basically found that these span the entire available environment in the room. So not only was a single location represented by a similar resolution, but all the different locations were represented by a similar number of firing fields and a similar resolution. So that was the previous work of what we have done with the bats. And as I mentioned before, what I plan to have in my lab, which we just started a couple of months ago, is studying the neural mechanisms of spatial behavior and social communication and vocal learning, which is erased over here. So I told you, I hope I managed to convince you a little bit that bats are a good model system to study spatial behavior, in particular, three-dimensional spatial behavior. But now I just want to make sure I drive home another point. So I often talk to people, and people think that, recording in 3D flying bats is probably the most uncontrolled experiment on the planet. They fly around, everything goes crazy in there. And I want to disprove that point and actually emphasize that this is one of the most controlled experiments that you can do on the planet. Because when the bats navigate in our dark rooms, they cannot use vision, olfaction, somatosensation. They have just one single sensory channel that they can use to negotiate that environment, and that is their sense of echolocation. And we can measure that sense at microsecond resolution and at very fine spatial precision. So I just want to, I don't know how active, I just want to show you this movie for a second. So what you're going to see here is going to be a bat flying in, and it needs to detect a target, which is located over here. And here are basically just simple microphones, ultrasonic microphones that track the bat echolocation. So you're going to see the bat flying here. Here's the target, and you're going to see its echolocation in a second now. So they basically fly. These particular bats have a double click. They go left, right, right, left. At some point, when they detect the target, they lock onto it, and then they start basically flying in a very straight trajectory towards that particular flight room. So we can, and another thing that I want to mention is that not only can we monitor every sensory beat coming into the system, but we can also control it very easily. There's nothing easier than basically playing noise back to the animal and jamming its entire echolocation. One more movie. And then the next thing I want to show is that not only can you monitor the sensory behavior of the animal, but you can also monitor the motor pattern of this animal at very fine resolution. So this is a system that we're now also getting for our lab. What you're going to see here on the right is going to be just a live feed of the bat that is flying around. This bat has 5 infrared markers, 1 on its head, 2 on the tips of its ears, and 2 on the tips of its wings. And then what you're going to see here on the left is going to be the reconstruction of all of those points. So as the bat flies around, using an array much larger than what I used before, it is about of 10 to 20 video cameras, you can reconstruct with very fine resolution the entire motor behavior of the bat as it flies around in a room. And you can do this reconstruction at distances of about even up to 10 meters easily. So this is just a point I want to take this opportunity to emphasize. This is not some crazy, uncontrolled experiments, but you can actually do it in a highly controlled way. The last thing I'm going to mention in just a second is going to be just the new direction of the lab, and again, just the motivation of why we're going in that direction. So many people know bats for their echolocation ability. They can navigate in the night using echolocation. What many people do not know is that bats also possess one of the most diverse and sophisticated acoustic communication repertoires on our planet. So these guys have an acoustic vocabulary, here just showing about 6 communication calls, which rivals that of higher order non-human chimpanzees. And they use this communication to live in a very complex social context. They live about 25 years, these guys in environments of hundreds of thousands of animals together. And so they developed this very rich acoustic and social behavior. And we can use those animals now to set the neural basis of acoustic communication. Another key aspect that we're going for now with respect to the bat acoustic communication is their vocal learning abilities. So while we learn vocally our language very, very easily, the only other mammals that exist on this planet and learn the language are basically elephants, cetaceans, meaning dolphins and whales, and bats. For very simple reason, the mammalian neural basis of vocal learning was never studied in the history of science. We only use songbirds, which, let me just emphasize, if I would not be working on bats, there would be only one model system on which I would be working on, and that is the songbird. It's a beautiful model system. There is just one caveat. It's not a mammalian brain. So here we have this, now that we have those tools to record from bats, we're now going into optogenetics and imaging as well in the bats. We have those tools to do a detailed investigation of mammalian circuits in bats. We also have this rare opportunity to study for the first time the neural basis of vocal learning in the mammalian brain. And so with that, I would like to conclude. I want to thank my former mentor, Nahum Lanovsky, with all the work that I described today was done. I didn't mention my work in a postdoc with Carlos Brody. If you want, I'll be happy to mention this is now in review, working on rats, acquiring many, many techniques, which are basically all going to be transferred now to the bats on accumulation of evidence in the basal ganglia. But this was a great period of time, and I want to thank him for that as well. And I've talked to some of you about this work. I'll be happy to talk afterwards about that as well. And thank you all very much again for the invitation. It's a great honor and a pleasure to be here and see what you guys are doing. It's amazing. Thank you.
Well, and thank you very much, Michael. That, just like I said, it was really delightful, wasn't it? So we have time for some questions.
Yep.
So Theta is clearly not doing those oscillatory interference models. So and you've shown that it's not related to running speed and all these other things that people kind of thought it was. What do you think its purpose is it's serving in those intermittent bouts?
No, thank you for the question. That's a great question. So, yeah, I just want to emphasize, theta oscillations in the bat exist in exactly the same way as they exist in primates and humans. So rats and rodents are, in fact, the highly unusual model system because they have these weird continuous oscillations. And, it was linked to spatial navigation. The only thing we have shown so far is that those oscillations are not necessary for the existence of place cells and grid cells, but there is a whole literature in humans about what those theta bouts can be good for, such as encoding new memories, for instance, working memory, solving complex cognitive tasks. These are all the things that people have shown to be potential candidates for function of theta in primates and in humans. For example, theta oscillations is about 120 milliseconds, 8 hertz, right? It's an effective mechanism for LTP, for binding new events together. So I think these, building on that literature now is a great, kind of direction that I think the field will be going now, trying to understand what theta oscillations are actually doing in bats and in rats as well. Yes?
I was intrigued by your teaser observations about vocal communication. Can you say a few words more about the two things? One, is it strongly biased towards when they're not flying, or does it occur also when they're flying? And secondly, are there individual signatures that allow bats to recognize one another?
That's two great questions, yeah. So first of all, bats use acoustic communication. Some species of bats, there are over 1,000 species of bats, so each animal has its own different ethology and they use it differently. But bats generally use acoustic communication for both. They also use them when they fly to coordinate group activity. So there's a very, we study a rat in a box or a bat in a flight room, but that's not the way we navigate. We navigate in social context. Bats do the same. They use acoustic communication to coordinate group activity. That's a different aspect we want to go into as well. But when they are in their caves, for the other half of their life, when they're in the cave, they use acoustic communication primarily for social interactions. And so now one of the things we're actively doing now in the lab is characterizing at a very great detail the exact social, behavioral conditions under which they communicate, how that changes seasonally when the animals are in different contexts, females versus male, juvenile versus adults. So we're doing all of that as well. The second question was, sorry, say again? Individual, yeah. So we don't know yet. So we're looking into this. We know, for example, that mothers can identify the unique isolation calls of pups. I just want to show this again. So when the bats are born initially, they're born with one single innate call. This is this call, which is a pup isolation calls, and mothers can basically detect those calls and then go and find their pup. And then with the course of learning, after about 90 days, these calls completely vanish from the bat repertoire, and they're replaced with these over 22 different types of communication calls which arise through learning. So yeah, but we don't know if at the adult level whether they can use communication or there's like unique signatures in their calls, but yeah, we're looking into this right now. Yeah.
Really quickly, can I just have the lightning talk presenters come line up with Angie, and then we'll have our next question up in the back.
Great question.
Can you hear me?
Yeah, I was.
So I had
Sorry.
a question regarding your. I'm over here.
Yes.
Your claims about theta being exactly the same as in rodents, in the bats, but essentially you're using tetrodes, right? So what you really have is one time series and a power spectrum, and that's it.
Yeah.
So I was wondering if you have, I mean, obviously, theta is not just a frequency, it's a mechanism that comes with sinks and sources.
Yeah.
Right? And so I was wondering whether there's a particular reason why you haven't tried depth recordings to actually come close, to comparing one-to-one to rodents in terms of theta, as well as the sharp wave ripples, as far as I can tell. You also showed some of that.
Yeah.
And whether you have any plans to do so.
So this has already been done. So my former mentor, Nachum, in his postdoc, he had a Nature Neuroscience paper. He did use tetrodes, but he basically moved them along, and so he did the whole analysis. It's similar to the rodents, and he found a similar mechanism. We have not done this depth recording with multiple points along the shank, and that's something I think that I agree with you. It should be done, and it should be done, and it will be done. Yeah. Yes.
So yeah, I had a quick question. In regards to the grid, place, and border cells, does the volume of the area described for those cells change with context, meaning flying versus crawling? Is it different for species, like between the rat and the bat?
Yeah, that's a good question. So yeah, we know even from rodent studies early on that if you take an animal and you have a place cell when the animal is in this box, and now you move it to this box, place fields are going to become larger. And we basically saw the same thing. So when I was recording place cells in a crawling bat, you get very small place fields. When the bat was flying around in a 6 by 5 by 3 meter flight room, the average size of the place fields was about 1 meter. In a revision of the paper, we had to do some more experiments in a smaller flight enclosure, and you see a slight reduction in the size of the field. So yeah, the size of the fields does change. It wasn't the same cell, but we did see systematic differences in the size of the fields that we were recording between the different environments. Yeah.
Story?
So I'm sorry, over here.
Yes.
This isn't really a science question so much as what kind of bats are you going to be studying? Because in the States, there's a bat epidemic.
Which is why we don't study bats in the States.
Okay.
Yeah.
I mean, but at Berkeley, are you going to be studying fruit bats from
Yep.
Israel?
Yeah, so we already have, if I had my computer, I could show our colony. We have hundreds of bats in our colony in Berkeley in the basement, and they live happily ever after. They breed very well, and they're acclimating very well. So we have the same
Great.
Egyptian fruit
I
bats.
feel much better. I have to put that in your grant proposal that you're using bats that are going to be okay.
Well, yeah, they're doing so far, they're doing very well, yes.
Peter.
You've already done a fantastic job solving this wireless recording problem in bats. So I'm curious, what are some of the remaining technical challenges that you expect to have to solve in the next years to work with bats?
Yeah, good question. So one thing is that now we no longer actually, system we had in the science paper is no longer used, in fact. So now it's all switching to data loggers. And the nice thing about data loggers, you basically can record literally now from an infinite amount of animals. I can record from 100 animals if I would like to simultaneously. And it's very easy to scale in number of channels. So that's one of the things we're working on now, basically scaling up in number of channels. We're now starting to develop, I could show you, I don't have the slide here, optogenetics in bats. So we know already the same viruses, exact viruses that work in bats, in rats, like Chernodopsin and GCAM6F that we tested, expressed beautifully in the bat. So we're going into optogenetics and, of course, wireless optogenetics. We're now going to calcium imaging in the bats, and we want to make it wireless. So we hope in a few years to image from 3D flying bats. So yeah, these are the minor technological challenges we need to face. But yeah, that's, I'm in the Department of Bioengineering and Neuroscience, and I'm a bioengineer, so I love those stuff. But yeah, these are the key directions we're going in right now into the technology. Yeah.
I don't mean to stop the question, but let's hear it for wireless telemetry of calcium imaging. Okay, so we do have a couple more minutes, and we can still stay on schedule. Yeah.
Great talk. Have you done anything with the sharp wave ripple data? Meaning, have you looked at the multi-unit envelope? Have you looked at structured reactivation?
Yeah. So we have not. What we have done, we saw that you have a very strong correlated activity of neurons to sharp waves. So we've shown that very clearly. The reactivation of patterns, we have not done that, and this is why we moved to data loggers, because with our older system, we could record from one tetrode at a time. So we only had 4 channels we can record on so many neurons. Now that we scale up with the number of neurons, we can basically have many, ideally hundreds of neurons recorded simultaneously in flying bats. And so then you can begin addressing this kind of questions. Yep.
Well, I'll ask a question then.
Yeah.
Obviously, you're interested in the bat language issue. So what kind of ideas are out there, beyond identifying individuals, about what the bats are actually talking about?
What are they talking about?
Yeah.
Well, just to emphasize one thing is that we don't study bat communication to understand bat communication. We study bat communication to understand a mammalian mechanism of acoustic communication between individuals like we're talking. And one of the amazing things is that there's nobody in the history of science has ever recorded from 2 brains simultaneously during a conversation, ever. So we don't know this mechanism.
You're going to do that while they're flying.
And that's the first level experiment for us, because the challenge is, of course, you need to fulfill 2 main requirements. One, you need a mammal, ideally, that communicates in a very interesting way. So mice and rats don't really do that. You can do it in primates, but then implanting multiple primates together and having them walk around freely and communicating socially is going to be challenging getting through the committees. But that's actually, for example, one very simple experiment that you can do and answer a major question. How do brains, for example, become synchronized during a communication? We know bats, we're now looking into this. When they talk to each other, you just put 2 bats together and they start just talk to each other continuously. I can show you if you want afterwards, just a conversation between the bats. They just talk to each other continuously back and forward. And that's a completely natural behavior of those animals. And we can basically now start looking at the neural basis of a conversation between multiple mammalian brains.
Do you think they tell the truth, or do they lie to each other?
I think they're honest, yeah, I know.
Maybe they're talking about where the good fruit trees aren't, or something
maybe
like
they're talking about maybe they're talking about us. I don't know. Yeah, absolutely.
Yeah. Okay, Anybody else? Yeah.
Can you say anything yet about the organization of auditory cortex? I'm mindful of the Suga's elegant studies, but focus on a narrow range of frequency representation, and now you're seeing, at least in some species, a much broader representation. Do you know whether there are specialized areas or components of primary and higher areas that are devoted towards these communication rather than echolocation domains?
That's a great question. So now the answer is no, but that's exactly what we're doing right now. So we're taking 2 approaches. One is the molecular approach. So we're looking for markers using immunohistochemistry. We're doing kind of IEG experiments, trying to identify the areas that are basically activating during acoustic communication, social communication, just hearing, just talking. So that's exactly what we're doing now. The second approach is tracing. So there's specific nuclei which are easily identified, like the ventral MGB, and we can basically go there and start tracing all the areas of the auditory cortex as well. So these are the two main approaches we're working on right now. Beyond that, I want to emphasize this is not my work. This was done in Nahum's lab. But actually, there is people, I think, we stick the electrode in the brain and we don't really know where it is. There is a very detailed brain atlas specifically for these bat species with the auditory cortex, hippocampus, of course, all other areas of the brain very well delineated, but now we're refining it even further for the specific questions and areas that we want to target. Yeah.
Okay, one more
Sure.
question.
Sure.
You said there's thousands of bat species.
Yeah.
So do any of them have brood sizes and generation times that suggest they might be tractable to genetic methods as we have known and loved them?
Absolutely, yes, absolutely, yeah. So, that's one of the reasons why I actually went to Berkeley is because CRISPR-Cas9 was originated in Berkeley, and that's one of the goals that we have for the next few years to basically introduce CRISPR into these guys as well. And that's another reason, as you mentioned before, why we chose those bats. The American bats actually are not very happy in captivity, so they don't breed very well in captivity, they don't prosper in captivity. These guys breed on the first day that we brought them into the lab. And so because they breed so well, and we can basically monitor the gestation and all of that, we can now begin to also introduce. So a CRISPR bat is not our first project, but it is definitely a project that we're going to be going for very aggressively in the next few years. Yeah, absolutely.
Wow, well, thank you very much, Mike.
Absolutely.